So let me give a brief introduction of Dr. Yufan. Dr. Yufan is a senior research scientist in the NVIDIA Moonlight team. His research interests include medical image segmentation, auto machine learning under foundation models. He obtained his PhD from GHU and was a recipient of Mikai Young Scientist Award. Moonlight has been one of the most impactful repositories in medical image analysis. Every time I read its code, it always gives me a sense of beauty. So thank you so much for your very remarkable contribution to the community. We are honored to have you here to kick off the algorithm presentation session.
Okay. Yeah, thanks, Madrin. Thanks for organizing this wonderful challenge and invite all of us here to discuss this topic. Hello, everyone. My name is Yufan. Today, I'm going to present our work called VISTA3D, a unified segmentation foundation model for 3D medical imaging.
Sorry for interruption. Could you please
Yes.
switch your slide to presentation mode?
Okay. Yeah, sure. Okay.
Okay, perfect.
So we're from the MANI team, the Medical Open Network for Artificial Intelligence, and the Madrin also already introduced this MANI framework. And beyond this framework, our team also have several recent publications about the foundation model. The first is the VISTA3D, and we also have other works like the diffusion and generative foundation model, the MACI, the medical AI for synthetic imaging. Also, we are working on the vision language model. We have the VILA M3. Uh, this is some recent works for our team. So get back to the Vista 3D. So when we designed the Vista 3D, the whole goal is to build easy to use workflow that cover most advancements in foundation models and they can actually save annotation effort. So the goal is not just an interactive model, it's trying to unify the advancements in all the foundation models. And our solution is trying to develop a unified model that supports accurate automatic segmentation for most classes and 3D interactive annotations with zero-shot ability. So the actual workflow for this model is like when people give you a 3D CT um, and tell the model to segment some class X. If this class X, from if it's lung, liver, or those common organs, this has a lots of models that already solve this problem using automatic segmentation. So there's no need for interactive interactions because interactive segmentation takes human effort. For most classes, if we have enough training data, we want the model to perform accurate or state-of-the-art automatic segmentation to solve this problem. And we introduced this human in the loop. So if you have this automatic segmentation, if this class X it belongs to some common classes like lung, liver, and the Visa 3D do automatic segmentation. And the humans can take a look at the results and doing some interactive refinement. This is the one loop. So if this class X belongs to some novel tumors or this is from something new, and the model has never been trained on it and it's not able to perform automatic segmentation. It goes to this right loop. It's trying to do the zero-shot interactive segmentation, which is based on the SAM work. It directly do this interactive zero-shot and it gets this human in the loop. So this is the whole design workflow for Visa 3D. So it's trying to reduce the human effort for the segmentation task. And so what we did is the Visa 3D supports diverse classes. It supports 127 classes, including 7 types of tumors and lesions. This is for the automatic segmentation. Also, it's trained on 12,000 CT scans on 25 public datasets. One limitation with Visa 3D is only trained on CT because we have to make sure it's all the datasets are commercial friendly license. This is for the automatic segmentation. Also, we support the 3D interactive segmentation. We have a very efficient 3D interactive segmentation ad-hitting pipeline. User can provide clicks to segment the 127 classes or refine this automatic segmentations. Also, the model was trained to enable the zero-shot capabilities. We can use clicks to segment unseen structures. For accuracy benchmarks, so we compare it with Auto3Seg and UNet and those segmentator on very diverse benchmarks. We also compare it to other foundation models like SAM, MatSAM, and the SegVol. And everything for this model is open source. It's under the Manai repository. All the model weights, training, evaluation, and dataset curation codes are open source. And it's integrated into Manai 1.4 and the Manai Model 2. It's very easy to deploy. The paper code and demo, it's all in this link. So here is the model architecture design. We have a dual branch design with the 3D SAM heads. So the whole framework is built on the traditional 3D segmentation pipelines, which is based on sliding window inference and the patch base. So we crop a patch from the 3D volume, and we use a 3D encoder decoder. This can be anything. Here we just use the UNet based. It's called SegResNet, which is basically just 3D encoder, 3D decoder with those long skip connections. We found in most cases this kind of architecture works the best. You can definitely add some transformer components to it. But in our case, we just use a convolutional unit, which is called SegResNet. After getting the feature maps from these two branches, then we have these two heads to do the segmentation. This is for the automatic segmentation, and this is for the interactive segmentation. For the automatic head, it's called auto head. When a user just provide a class prompt saying we want to segment liver or whatever, it's just an index. You can some people also try to use text prompt. We also try this, but I thought we found that open vocabulary segmentation is pretty hard. So we just directly give them an integer index. Like liver is 1, spleen is 2, just integer indexes. And then it extracts the embedding from our predefined embedding space and just do MLP and linear projection to project this feature map into the final segmentation. And our auto had achieved a similar performance to the segmentator. This is for the auto part. The other part is called the point head. The point head is we follow the SAM design, but we changed everything to 3D. So this is a 3D feature map, and we downsampled it a bit so the memory can be we can work on the model can be run on smaller GPUs. And then the point prompt is 3D. We just follow the SAM design, convert everything to 3D. And we have this transformers across attention and finally get the interactive results using the point clicks. And then these two results are merged using some focal click kind of algorithm. We use these interactive results to correct the automatic results. This is the whole pipeline. Also, if you don't provide the auto, so we don't do auto segmentation, we can purely doing the interactive segmentation, which can be used for zero-shot segmentation. This is the dataset curation. We curated on over 11,000 CT scans with partial labels, pseudo labels, and super voxels. The partial labels, which is similar to this challenges dataset, which is the manual ground truth from each dataset. We mapped all those segmentations to a global index because some data has liver as index 1, some data has liver as index 10. So we have a global index and map everything to this global index. So we can do automatic segmentation, training on all those partial label dataset. The other thing we do is pseudo labels. We generate lots of pseudo labels from some total segmentator model and to improve the robustness. And the third is super voxels. I think we found the super voxels. Lots of people are also using super voxels. It's very important to improve the generalizability of the zero shot, the zero shot ability. We use the 3D slick super voxel from 3View SAM features, and the result looks like this. So we use all the three partial labels, pseudo labels, and super voxels as the ground truths and combine them together to train this interactive segmentation and automatic segmentation. And this is the data set we used. This is for the model itself. For the training recipe for this model, it's pretty complicated 4-stage because we have to train both automatic segmentation and the point interactive segmentation. Also, we have 7 tumor types to segment, and we have this huge class imbalance issue because for lung tumors only have 60. But for lung or liver itself, we have like 10,000 scans. So so we split this into 4 stages to solve the two branch conflicts, data imbalance, and the performance issues. And what we did, it's pretty complicated. We just trained the point branch first with supervoxel, pseudo label, and partial labels. This is just trying to train a very strong encoder. This step will result in a state-of-art interactive model. We just train this branch because this branch doesn't need to do automatic segmentation. And then we fine-tune the point branch with underrepresented data set, like some tumor classes, to solve this data imbalance issue. After that, we just freeze this point branch and freeze the encoder, and we train this automatic branch and train this upper bar. This is just using the traditional automatic segmentation recipes. And the last step, we just fine tune the last branch, the auto branch with underrepresented classes for tumors to improve the results. So the whole recipe is trying to push the performance and solve the conflict between two branches, which is too much complicated for this challenge. And so what we did is we simplified the whole framework and to just support the interactive segmentation using this branch. So the participant can completely ignore this automatic part and just focus this part. So if you focus this part, it's the framework becomes super simple. It's just a encoder decoders UNet to extract features, and the features attached with a 3D SAM head to perform the interactive segmentation. This is for this challenge. So we put this the simplified code into this repository. So for how to use, I would just show the code directly. So this is the VISTA3D repo, and we have 3D and 3D. So for 3D, we have this is the README. This is the full README for the code base, and we have a simplified version called the CVPR workshop. And if you go to it, we only have 2 scripts called train and infer. This train, it's so for installation, it's just a pip install. For training, it's already have the multi-GPU supported, just torch run the train CVPR, the Python. And for inference, you can use the Docker, or you can directly run this infer CVPR. That's 2 scripts you can use. For train CVPR, I think we also simplified a lot. We have this is some login to write to TensorBoard. You can use it, and this is the dataset. This is just dataset. You just change the base path to your dataset, and you provide a JSON file that includes all your data, image, and the label. And this is very easy to modify. Also, once you load the image from the NumPy MPZ files, I will give this A5 matrix, and you can use the Manai transforms to pre-process like scale intensity, spatial pad, random crop. So this Manai transform actually provides you all this transformations you can use. And the final output is just the data, the image and the label pairs with all the augmentations. And during training, you just set some hyperparameters here. And below the checkpoints, I already fine-tuned the VISTA3D on this 10% subset. I think it's in the README. You can download that. We already fine-tuned the VISTA3D on it. And then I think all these are very pretty standard PyTorch code. And here the model itself, it's already in Manai 1.4, so you can directly import the VISTA3D 1.32 into it. If you want to modify the architecture itself, you just go to Manai networks, and the VISTA3D 132 is defined here, Manai networks next VISTA3D. You just copy this file into this repo, and you can make modifications here. Basically, this is just a backbone, which is in ResNet, then 2 head, the point head and class head, and that's it. You can modify here. Everything is in this Python file. This is model. If you don't need to modify the model architecture, you just import this, and everything else, optimizer, learning scheduler, and yeah, that's basically. So for training, when I start the iteration, we provide the sample prompt pairs. This is also from Manai. You can use this, and it will give you all the necessary train outputs you need to train this interactive segmentation. They give you the point, which is the point clicks, the point label, which means if it's foreground or background. The prompt class give you the additional information about this point, whether it's a liver, it's a spleen, or whatever. And label prompt also tells you if it's a liver or spleen, so you can use it to train automatic segmentation or other techniques. So the skip update is
Sorry, Yufan, sorry for interruption. Yeah, we are a bit out of time.
okay. Yeah, I'll finish quick. Okay. So this is the train, which is pretty standard. The infer is also standard. You can convert clicks to standard format, and yeah, I think one thing you need to notice is we provide this point-based window infer. This so VISTA3D inference is when you provide the click points, we just crop a 128 qubit patch around this point, and then we only perform inference on this patch. And that's it. I think just these two scripts, it's very easy to read. Let's see what else. Okay, the potential improvements. I think you can do iterative point sampling just like the validation. The original VISTA3D provides this script. You can look at that. Zoom in, zoom out, varying window size for different modalities. I think Interactive and the SegVol, they already use this technique, so we can learn from them. Model-specific augmentations, normalized reorientation for all different modalities. And also you can use bounding box to limit the response area. VISTA3D currently only support the point clicks. So but you can use bounding box to refine the final results.